Greetings, and a good day everyone. To fight any pathogen with rational means, it's important to understand how it operates and which resources it needs from its environment. In the case of microcellular human pathogens, this environment is the human body. That's why knowing the molecular machinery the pathogen needs to interface with host cells or fluids can help guide therapy and develop drugs against its most vital targets. In this presentation, I will try to answer the question, what do pathogens need from the host? I will begin by exploring a more broader question, what does life need in general? And how do the archetypical lifestyles of organisms determine their basic needs? Then I will go into more detail about a specific agent of disease and its intricacies. These will include general information about its physical attributes, habitat, epidemiology, and disease, as well as the vital genes, proteins, and other mechanisms the pathogen needs to live and thrive in its niche. Lastly, I will talk about the ways the pathogen has to avoid our defenses and establish infection. Afterwards, I will look at what we can do once it happens, or in some cases, even prevent infection entirely. The efforts that have been made to discover effective drugs, and how they work. Same for vaccinations, and if there is one, where it is applied. Now I will begin with the most general question. What does biological life, as we know it, require to function and proliferate? The most basic answer to this question is CHNOPS, or in other words carbon, hydrogen, nitrogen, oxygen, phosphorus and sulfur. The first four form all basic building blocks of information storage, the nucleosides. Add phosphorus to that, and you have nucleotides, able to form long polymers through the ribose phosphate backbone, and the basis of all information storage in life as we know it, as well as the main components of most biological membranes, lipids and phospholipids. With a further sulfur atom, important ingredient to disulfide bonds, we can produce every amino acid except for seleноcysteine. Of course, the devil is in the details. Beside these six main components, there's a plethora of trace elements, such as iron, magnesium, zinc, sodium, potassium, chlorine, and many others, which are nonetheless vital for many enzymes, such as the DNA polymerase or adhesion molecules, pH or electrolyte balance, or other functions in the catalytic centers of enzymes. For example, here's the makeup of the human body. As we can see, it's mostly water, protein, and fat. In terms of elements, oxygen reigns supreme, after the carbon, hydrogen and nitrogen, the basic building blocks, but also a surprisingly large percentage of calcium, probably from bones and the electrolyte balance. Some elements can also replace others, such as arsenic instead of phosphorus. On the other hand, this similarity can also lead to toxic effects if the organism isn't adapted to a certain concentration. Otherwise, iron also plays a major role, not just for our red blood cells, but also most bacteria. So much so that the ability to uptake iron from the environment is one of the limiting factors of bacterial growth. In a healthy body, the levels of free iron are so low that most bacteria can't grow, and secreting more lactoferrin to reduce the levels even further is actually one of the immune responses during infection. Even though all life we know needs the same building blocks, the way they procure them varies vastly, so it makes sense to split them into different groups. These are the autotrophs, organisms which are able to produce organic substances and nutrients such as sugars and proteins from inorganic compounds like water, carbon dioxide and ammonia. Plants or cyanobacteria come to mind first, since they mostly use these inorganic substances and sunlight as their energy source. They are photoautotrophous. However, there also exist chemoautotrophous organisms which use other chemicals as energy source, such as the inhabitants of deep-sea black smokers. The second group are the heterotrophs, all other organisms which, in some way, must feed upon others, be they dead or alive. For the purpose of this presentation, I will mostly focus on those that take from the living without giving anything in return. The parasites. Among the parasites, there are again different classes. I won't cover viruses, because they can be hardly considered alive outside the host, and once inside, they mostly use the cellular machinery. Some can only live inside host cells, usually because they lack the means to produce vital molecules themselves. Accordingly, they also tend to have a smaller than normal genome size, because they lost the unnecessary genes over time. Good examples are chlamydia, Coxiella and Rickettsia. It's also very difficult or impossible to grow them in culture, which can complicate diagnosis. Then there are those that can proliferate with inside and outside of cells, such as Histoplasma capsulatum, Legionella or Listeria monocytogenes. They have special genes they can activate once within the body, which then allow the invasion of host cells, persistence, and immune evasion. More on that later. And there are also pathogens which might not cause disease in healthy individuals, but proliferate and take over once the circumstances are right, or the immune system is compromised. Common examples are intestinal infection with Clostridium difficile, especially after treatment with broad-spectrum antibiotics, catheter infection or sepsis from skin flora bacteria such as Staphylococcus aureus or epidermitis, and secondary infections after a primary lung infection like Streptococcus pneumoniae or Haemophilus influenza, which can also be found around the throat of healthy individuals. The human body is much more dense in nutrients than most natural environments, so a pathogen which knows how to take advantage of that can proliferate rapidly. That's where we come to resource management. Transport of small molecules across the membrane is mostly facilitated through channels in the cellular membrane, or special proteins that are secreted, can bind nutrients with a certain affinity, and are then taken up again. Both transports can be passive or active. Here we can see that the use of carrier proteins has a clear advantage in settings of low or medium nutrient concentration. At high concentrations, the carrier proteins become saturated, but until then, even a relatively small amount of carrier proteins can facilitate the uptake of a much larger amount of nutrients than simple diffusion could. Examples for channels and carrier proteins in bacteria include the beta-galactose permease, one of the proteins under control of the Lac operon and encoded by the gene LAC-Y in E. coli. FBPA, or ferric binding protein A, is a transferrin protein from Neisseria gonorrhea, which shuttles iron from human lactoferrin carrier proteins into the bacterium, and is a virulence factor. Another important aspect of bacterial extracellular pathogens and virulence factor is the formation of biofilms. It provides a formidable defense against immune defenses and drug therapy, and yet it's able to sustain the bacteria within. The highest risk stems from biofilms on foreign objects within the body, such as catheters, implants, or artificial heart valves. Even now, replacement of the colonized part more often than not is the best course of treatment. The biofilm consists of polymeric secretions, such as polysaccharides, proteins, and DNA, and due to their viscous nature, diffusion through it is very slow. Which begs the question, how come the bacteria inside don't starve? As it turns out, there may be channels within the biofilm, naturally forming as it develops. At least they have been found for Bacillus subtilis on agar medium so far. Here we can see a green dye flowing through the channels, 2 scanning electron microscope images in black and white, and a model schematic of the structure. Other researchers found that, in a computer model, the motion of many small motile bacteria in a colony added up and formed a large-scale fluid vortex, which could also support nutrient distribution, even when the individual bacteria were randomly oriented. For the sake of this presentation, I will be focusing on the pathogen Borrelia burgdorferi. It's the causative agent of Lyme borreliosis. The disease manifests itself as a painless, circular red rash around the center bite mark of a tick after a short time. However, the long-term consequences can be much more severe, like muscle and joint pains, inflammation, and even problems with the nervous system or irregular heartbeat. The tick that causes the disease, Ixodes, can be found in America, at the Atlantic or Pacific coast of the United States, in parts of Western Europe and Eurasia, and dogs and birds can also carry the bacterium. Up to 20% of ticks found in endemic areas may be vectors of the pathogen. And speaking of which, the pathogen itself is a gram-negative spirochete with a very characteristic shape. It's rather long, but only has a diameter of 0.3 micrometers. Its corkscrew shape and intracellular flagella allow it to swim through highly viscous fluids, like the space of the interstitial matrix within the human body. It can even swim forwards or backwards, since it has 2 flagellomotor protein complexes at each end of its body. This allows it to circumvent obstacles, and even evade macrophages. And here we can see that it has a rather complex membrane structure, with the 2 double membranes and peptidoglycan layer of gram-negative bacteria, a space in between for the flagella, and different membrane-bound, membrane, integral or transmembrane proteins. The OSP surface protein, in particular, will be important for later. During the tick bite and infection, Borrelia changes its outer surface protein, but within the tick gut, it's always OSPA. The ABC transporter system is also important for infection. Once the tick sucks blood, the chemical environment of its gut changes. Borrelia burgdorferi can sense these changes and reacts by secreting a signal molecule called autoinducer 2. This is taken up by the ABC transporter and allows for quorum sensing. Only when enough signal molecule from all the individual Borrelia bacteria is present, they decide to move to the next stage of their life cycle. They switch the expression of OSP from A to C, which lets them migrate from the tick into the human host and bind to fibronectin. This leads us to the next topic, the immune reaction of the host, and how Borrelia manages to avoid it. One of the quickest and most effective early immune defenses against bacteria is the innate immune system and the complement system. Since gram-negative bacteria don't have the very thick peptidoglycan layer, one might expect Borrelia to be lysed by the complement cascade, starting with C3 and ending with the membrane attack complex. However, Borrelia has its own defense against the defense, in the form of CRAS proteins on the outer surface. These bind surrounding molecules from the extracellular matrix and form a protective shield around the bacterium. What about the general Achilles heel of bacterial growth? Iron restriction. Well, Borrelia got very creative. Unlike most, it does contain hardly any iron at all, and used manganese instead. It's estimated that a single cell only contains 10 iron atoms on average, and simply doesn't need them for enzymatic function. But surely at least the adaptive immune system can take care of this pathogen? As it turns out, even specific antibodies have a hard time targeting Borrelia, because it can change its outer surface proteins, similar to Leishmania parasites. These immune evasion strategies, and others, allow Borrelia to persist for months or even years in the body if the immune system doesn't react in the right way. A correct response consists of a balance between Th1 and Th2. Without sufficient Th1, the immune system is not activated, and macrophages don't kill Borrelia efficiently enough so that they disseminate and spread through the body, and without Th2, the immune reaction persists for too long and causes chronic symptoms or autoimmune disorders. Now that we know how Borrelia persists, we can explore how it takes advantage of its host. It may not be an obligate intracellular pathogen, but it still is very dependent. For example, it has no genes involved in the TCA cycle or oxidative phosphorylation. So glycolysis is the only possible source of energy for the metabolism. This is inefficient in comparison, and might be one of the reasons why Borrelia grow very slowly. The 6 principal carbohydrates it can use as energy source are glucose, glycerol, maltose, mannose, N-acetylglucosamine, and chitobiose, which is an N-acetylglucosamine dimer. At least in vitro, trehalose was another possible energy source. Glucose can be taken up by the ABC transporter as well, glucose-6-phosphate or glycerol by another transferases. Also, it can't synthesize any fatty acids, amino acids or nucleotides. Accordingly, Borrelia needs to take up all these compounds from their surroundings. Its genome thereby contains over 50 genes for different kinds of transporter proteins and salvage pathways to ensure nutrient supply. For example, the genes BBB22 and BBB23 are known to encode for proteins facilitating transport of the purines hypoxanthine, adenine and guanine from surrounding medium into the cell, so-called purine permeases, and they are essential for the survival of Borrelia during the infection of mammal hosts. Now I want to move over to the third part of my presentation, the development of therapy and prevention of the disease associated with the pathogen. First, the detection. For the primary disease, this is usually done through an ELISA test for antibodies against Borrelia burgdorferi in serum. To keep false positives to a minimum, once ELISA is positive, a Western blot is performed to detect more antibodies against multiple different Borrelia antigens. The treatment then usually involves the antibiotics doxycycline, amoxicillin or cefuroxime, for 2 to 3 weeks, respectively. These inhibit the bacterial 30S ribosome subunit or the peptidoglycan synthesis, and are also used against other bacterial infections. Due to the low reproductive rate of Borrelia, treatment time may be longer than usual, but they can be given orally. If the infection reaches the central nervous system, intravenous antibiotics are the course of action. The causative agent of Lyme disease has only been identified in 1982, and no specific drugs are used so far. People simply tested known antibiotics and found that they worked. However, the more debilitating effects of the infection only manifest after the primary infection, and only in some. They are collected under the umbrella term of PTLDS, or post-treatment Lyme disease syndrome. Antibiotics do not help to alleviate these, It's assumed that they are caused by an autoimmune mechanism rather than the pathogen itself, and the fatigue can last for months or years, but usually gets better over time. There's no specific treatment, only therapy against the symptoms, such as pain relievers against joint pain and anti-inflammatory medication against the swelling. The best hope is to treat the primary infection as soon as possible, or avoid risk areas in the first place. And since prevention is preferable to therapy, we'll now explore the current vaccination options against Borrelia burgdorferi. There was a vaccine in 1998, called Lyme, which consisted of the OSPA surface protein with adjuvant, which was withdrawn from the market in 2002 because of poor sales and safety concerns. It actually blocked the initial transmission of the pathogen directly after the tick bite. However, required relatively high serum antibody titers to do so, which meant frequent booster vaccinations. The reason was that the antibodies in the blood had to be high enough before encountering the antigen, because once the antigen would activate the adaptive immune cells, it's too late to prevent the infection. Since Borrelia would switch from OSPA to OSPC, and there were also reports that the vaccine itself might cause autoimmune reactions, but it's unclear whether this really is the case. There have also been studies with vaccines targeting the predominant natural reservoir animal, a mouse species, which could be delivered orally in bait, and contains a modified vaccine virus expressing the OSPA protein. Others target not the pathogen, but rather the tick vector, by targeting enzymes that are necessary for the feeding on the host. However, none of these are currently implemented. This means that today, treatment after transmission and infection is the only course of action in humans. On the next slide, I'll show what might be coming up in the future. Currently, there are multiple studies and considerations. In one, mice were immunized with a life-attenuated Borrelia burgdorferi strain, and were thereafter protected from infection with the wild type. Others have tested protein vaccines with different proteins from the bacterium, or formulated the OSPA vaccine. And there have also been developments in terms of treatment. One research group found persistent peptidoglycan molecules in the synovial fluids of joints even after clearance, and assumed that this caused a long-term inflammation and pain, as confirmed in a mouse model. Drugs which degrade or otherwise remove the peptidoglycan might be a future treatment to prevent or shorten the post-treatment Lyme disease syndrome. And lastly, another study group found a slow-growing, dormant variant of Borrelia, resistant to standard single antibiotic treatment, that caused stronger joint inflammation than the normal, faster-growing variants. They assumed these to be the main causative agent of post-treatment Lyme disease syndrome. In a mouse model, a combination of the antibiotics daptomycin, doxycycline and ceftriaxone was able to eradicate this persistent form. This concludes my presentation on the question, what do pathogens need from the host? Here, I'll deliver the closing statements. We know that all biological organisms need the main components of the cell, carbohydrates as energy source or for structure, amino acids for proteins, structure, and enzymes, and nucleotides to store information and regulate the cell. Also, some proteins need ions for their function, and a specific pH range for stability. And these compounds can be taken up through more or less specific carrier proteins and membrane gates. Pinocytosis or phagocytosis are less specific options of bulk biomatter uptake. For pathogens, the rate at which they can gather necessary components determines how fast they can grow, and the human body uses, for example, iron restriction to prevent bacterial growth. Third, obligate intracellular or extracellular parasites need to acquire some compounds from their host to survive, and have, in some cases, lost the genes related to their synthesis entirely. And lastly, Borrelia burgdorferi as an example obligate parasite has many immune evasion strategies to work around the host defense and restriction. And here are my sources. Page 1 and page 2. Thank you for listening, and have a nice day.